Welcome everyone, and thank you for joining us for this last Agro Lunch and Learn webinar of 2021. This webinar series is made possible through the generous support from Eurofins AgroScience Services, so we thank them very much for their continuing sponsorship of this Lunch and Learn webinar series. My name is Laura McConnell, and I work for Bayer Crop Science in St. Louis, Missouri. I'll be serving as moderator for today's webinar. Attendees will be able to submit their questions and comments for the speaker via the Q&A tool. So if you notice, there's two different tools there. The Q&A, that is for questions for the speaker. The chat, if you have a technical issue and you need to reach out to our host, Kyle Sawyer, you can use that. So just remember to use the Q&A tool. So before I introduce our speaker, our distinguished speaker, I'll take a moment to highlight the benefits of becoming an Aggro member. Aggro pays enormous benefits by enhancing your science, building your scientific network, and supporting your professional development. So please go to the Aggro website at agrodiv.org and click on Become a Member. The cost is less than $20 a year, and you don't have to be a member of the American Chemical Society to be an affiliate member. You need your support. So I'd also like to remind you about the next Agro program, technical program, which will be happening next year in 2022 in Chicago in August 21st to the 25th. And right now we're accepting symposium proposals through November 15th. If you have any questions, you can contact our program chair, Heidi Ehrig. If you have a symposium idea, you're not quite sure, you can email her and discuss that with her. So thank you for that. So now I'm very proud and excited to introduce you to our speaker for today, Dr. Ralf Nauen. Ralf is an insect toxicologist, biochemist in R&D at Bayer Crop Science in Mannheim, Germany. He is a distinguished science fellow at Bayer, which is the highest level of fellow that you can be awarded from our company. He is also fellow of the Entomological Society of America, and he received the International Award for Research in Agrochemicals from Aggro back in 2014. He is author of more than 220 peer-reviewed articles and has received the highly cited researcher award by the Web of Science for the last 3 years. And he is also, in his spare time, in addition to his regular job, he is a university lecturer at the Agricultural and Molecular Entomology Department of the University of Bonn. In addition, he serves as executive editor of Pest Management Science and serves on the editorial board of many scientific journals. So without further ado, I'm going to pass it off to Ralf, and I'm going to stop sharing and let him get started on his presentation.
Okay, so I hope you can see my slide, the first one, as well as a laser pointer, and you can hear me.
Yes, we can. Thank you so much.
That's great. So Laura, thank you very much indeed for this kind introduction. And so it's a real pleasure for me to be invited by the organizers to share with you some of our work we did on bee toxicogenomics, and yeah, to be invited to this particular webinar. I wasn't aware it's the last of the year, so let's go ahead. So there's an important role of insect pollination. Just want to tell you that the worldwide economic value of insect pollination is immense, and that highlights that insect pollination is of vital importance in modern agriculture, especially considering crops such as, for example, apple, almonds, California almonds, melons, and strawberry. On the other hand, important staple food crops, such as, for example, wheat, rice, or corn, they do not rely on insect pollination. And so in total, 5 to 8% of the global crop production depends on insect pollination. Pollination of wild and cultivated plants is an indispensable service provided by a diverse range of free-living organisms and commercially managed bee species, as you can see here. And particularly the Western honeybee is the most important managed crop pollinator globally. So while global agriculture is expected to see an increase in pollination-dependent production, parts of the world are facing a decrease in bee abundance and diversity, raising concerns about the possible shortcoming of pollination services. There are over 20,000 different bee species in the world. Most of them are wild bee species and of course not managed pollinators. And roughly 2% of these species are involved in the pollination of 80% of crops worldwide. Honeybees are managed, artificially bred agricultural animals similar to livestock such as pigs and cows, I would say. So, and there are potential factors which contribute to insect pollinator decline or bee pollinator decline in particular, including, for example, parasites such as the varroa mite you see here sitting on the back of this honeybee, and also diseases which are transmitted, for example, by the varroa mite, such as the deformed wing virus you can see here. Other factors are climate change, habitat loss, diseases, and of course, pesticides are discussed heavily in the public. Particularly risks related to unintended bee pollinator exposure to insecticides applied by farmers to keep destructive crop pests under economic damage thresholds is of major concerns. Many insecticides are acutely toxic and have side effects on honeybees, especially those addressing neuronal target sites, such as, for example, voltage-gated or ligand-gated ion channels, which are known to be rather conserved among insects. And that's what you see here. So that is the overall insecticide market. And most of the insecticides act on nerve and muscle function. And a few chemical classes of insecticides address the nicotinic acetylcholine receptor, acetylcholinesterase, voltage-gated sonneschimmen, and the ranitidine receptor. So these are the most important target sites, and these are fast-acting insecticides. And most of these target sites are highly conserved among insects. So insects are diverse and have a complex evolutionary history. So honeybees diverged, for example, from Diptera and Lepidoptera approximately 300 million years ago. But developing insecticides selective to bee pollinators is an intrinsically difficult task because honeybees are insects. So it's not a surprise, at least for the entomologists amongst us, that insecticides also show side effect on honeybees because they are insects. So therefore, it's very important from a regulatory perspective to test new insecticides or also insecticides which are going to be re-registered for insect control in so-called acute toxicity tests. And that is a first-tier testing. It's tested by oral exposure as well as contact exposure. And here what we are testing is in fact we are looking at the LD50 value. And there's an EPA classification that all compounds which show LD50 values against honeybees in these tier 1 acute toxicity tests, which are lower than 2 microgram per bee, are considered highly toxic. Those between 2 and 11 microgram per bee are considered moderately toxic, and those which are showing LD50 values higher than 11 microgram are considered practically non-toxic. So considering the highly conserved target sites addressed by many chemical classes of insecticides, it's not a surprise that 95% of the top 20 insecticide by sales worldwide are acutely toxic to honeybees. So depending on the mode of action, dose and exposure route, pesticides can have lethal and sublethal effect on bees, which ultimately can impair their pollination services. Because of these potential effects, a risk assessment for bees is required by many regulatory agencies. and for the registration of these compounds. And that's what you see here. It's a tiered toxicity testing approach. It starts in the laboratory, goes into semi-field trials with tier 2 and field trials at the end. So, and that is an example taken here from the European Union. These assessments have traditionally focused primarily on honeybees, but have recently been extended to other bee species, such as, for example, the bumblebee or even red mason bees, which is a solitary wild bee species, because it has been shown that honeybees are not always a surrogate for other bee pollinators, which are used as managed bee pollinators or also wild bees. So there are diverse routes of exposure to crop protection products. And this is a picture taken from a very nice article from Reed Johnson, published in 2015. And you see there are many routes of exposure. It's overspray, it can be just guttation water, systemic pesticides are taken up by the plant. You find pesticides maybe in the water, in nectar, pollen, propolis, or even dust. And also other things are combined effects of multiple stressors, which might act synergistically, and which is also under close scrutiny at the moment, in particular for chemical mixtures. I will come to this later in my presentation. The honeybee genome has been sequenced many, many years ago, almost 15 years ago. It was one of the first insect genomes sequenced, and it was analyzed, and there was a satellite paper published at that time saying that there is a deficit of detoxification enzymes, and that the deficit of these detoxification genes in the honeybee will translate to less pesticide detoxification capability, which would then explain the species' unusual sensitivity to pesticides. I think the species is not unusually, I would say, sensitive to pesticide, and it has been shown later in a paper published by Hartstone and Scott when they analyzed 62 different insecticides. And they came to the conclusion in their paper that honeybees were not more sensitive than other insect species across those insecticides they excellent. I give you a few examples here. So honeybee selectivity is found in many chemical classes of insecticides, even classes of insecticides where you would not expect it. So this is one example you see here, Cumafos, an organophosphate, which is used as an in-hive medication to control varroa mites or smaller hive beetles. And chlorpyrifos, on the other hand, is also an organophosphate. And there is a difference in toxicity in honeybees between the two compounds, more than 200-fold. There's only 3-fold difference against the smaller hive beetle. So there is selectivity among organophosphate insecticides, which have been launched almost 80 years ago. For the pyrethroids, we can claim the same. We know pyrethroids, such as, for example, taufluvalinate, which are fairly safe to honeybees, because also taufluvalinate is a medication used to control varroa mites in honeybee hives. And you see here deltamethrin, which is another pyrethroid insecticide. Although the difference from a chemical perspective is not big between these two molecules, there is a high selectivity. So deltamethrin or taufluvalinate is almost 7,000-fold less active against honeybees compared to deltamethrin. Although the difference against a pest insect, in this case Helicobacter pyrgera, is almost none between these two compounds. So again, selectivity also for the pyrethroid insecticides. And this has been shown in a paper published more than 10 years ago by Mao and co-workers. And they claim that it's because of CYP9Q-mediated detoxification of acaricides in the honeybee, which make them safe. We picked this up, and we looked also into the neonicotinoid insecticides. As you know, neonicotinoids are under public debate, but you also find selectivity among neonicotinoid insecticides. Here's an example taken at thiacloprid versus imidacloprid. And thiacloprid is more than 2,000-fold less acutely toxic against honeybees compared to imidacloprid. Whereas the efficacy against one of the major target pests, in this case green peach aphid, there's almost no difference. Here, thiacloprid is even more active than imidacloprid. So what makes these honeybees tolerant against this neonicotinoid insecticide? It has been shown already in work published almost 20 years ago by Iwasa and colleagues. And they showed that there is differential toxicity of neonicotinoid insecticides in the honeybee. And I give you the example here. Here again, imidacloprid and the LD50 you can see over here versus thiacloprid and the LD50 over here. And they applied synergists which inhibit detoxification enzymes. In this particular case, synergists inhibiting cytochrome P450 enzymes. And they obtain synergistic ratios, which are quite similar here. So if you add an azole fungicide to thiacloprid, you make it more than 1,000-fold more toxic to honeybees. So that is an indication that detoxification enzymes are involved in the selectivity observed. We picked this up in a project which we call Toxicogenomics or bee toxicogenomics. And the aim of the project was to expand our understanding how insecticides work in bees and what makes bees different from pest insects. We also were interested in the identification of the molecular determinants of selectivity. And can we convert, at the end of the day, this knowledge into future products, make them safer per se? And we are looking also for an improved science-based communication with authorities. It's no longer enough to show that an insecticide is not active against honeybees, but the authorities, especially also in Europe, they want to know why are these compounds no longer active against honeybees and other bee pollinators. So as I mentioned earlier, bee pollinator safety of neonicotinoids is an ongoing public debate. Therefore, we have chosen this particular example. And neonicotinoid insecticides, for those of you not familiar with this class, are systemic insecticides. And they reversibly bind to insect nicotinic acetylcholine receptors, which are found in the central nervous system. These are receptors which are heteropentamers and ligand gated. They are gated by acetylcholine, which is a natural neurotransmitter. And neonicotinoid insecticides are applied in several ways as soil insecticides, by seed treatment, foliar, to control a broad range of sucking and several chewing insect pest species. And this is the IRAC, the Insecticide Resistance Action Committee Mode of Action Classification Scheme. And here you find these insecticides grouped in number 4, nicotinic acetylcholine receptor competitive modulators. We conducted radioligand binding studies using honeybee head membranes, and we found that thiacloprid and imidacloprid, they did not differ in their affinity to nicotinic acetylcholine receptors in the honeybee brain. So the selectivity definitely is not because of differences at the target site, which is highly conserved anyway between insects. Then we applied thiacloprid and imidacloprid to honeybees, and we checked for the parent compound by using UPLC MS-MS. And we found out that within 2 hours, thiacloprid was almost completely eliminated from the honeybee, whereas imidacloprid was not eliminated in that quick manner. And we looked for the major metabolite, and the major metabolite we identified was hydroxylated thiacloprid. Hydroxylation is the chief reaction involved in phase 1 metabolism of xenobiotics and catalyzed by cytochrome P450 monooxygenases. P450s are an enzyme supergene family and practically present in all living organisms, as you can see here. They require a cytochrome P450 reductase as a redox partner for electron transfer. P450s catalyze at least 60 chemically distinct reactions, and these are heme-thiolate enzymes, roughly 50 kilodalton. And insect genomes can carry between 36 and almost 200 P450 genes. Mosquitoes carry quite a lot of these P450 genes. The smallest arthropod cypome consists of 23 genes. It's that of the tomato russet mite. Insects have 4 major clans. That's how they are called. What you can see here in this graphics. It's the CYP2, CYP3, CYP4, and the mito clan. And 2 minor clans only very recently described, CYP16 and CYP20. It's different from plants and vertebrates, which have 11 clans. The major expression sites of P450s in insects are the fat body, which is the equivalent to the human liver, if you want, or the vertebrate liver, the gut, and the Malpighian tubules, which is the equivalent to vertebrate kidneys. And these P450s are known to confer insecticide resistance in pests by isoforms, which are duplicated or upregulated. The nomenclature of P450s is quite simple. Here you have the gene identifier, cytochrome P450, CYP, and then you have the family, which is shown by a number, and the subfamily shown by a letter. And all P450s belonging to the same subfamily have a homology or similarity of 55%. P450s are of major importance. And here you see the different phases of xenobiotic elimination, which could be involved in the detoxification of xenobiotics such as insecticides. P450s are those enzymes which functionalize these substrates, making them processable for other enzymes, such as involved in phase 2 in conjugation, such as, for example, UGTs or glutathione transferases. And these compounds are then conjugated with sugars or glutathione. And afterwards, they will be eliminated in phase 3 by so-called unidirectional efflux pumps, pumping them out of the cell. And it's the so-called ATP binding cassette transporters. All this is all this you find also in a honeybee. So honeybee are principally able to detoxify compounds, and of course they are also exposed to compounds such as, for example, floral nectar alkaloids, which they need to detoxify because they are quite toxic. The honeybee detoxome, that's how I call it, comprises 133 genes described in this paper here, published by Birnbaum and Johnson. In the current opinion of insect sciences in 2015. And they described 46 P450 genes. We did an experiment where we sequenced 48 honeybee transcriptomes, taken bees at different developmental stages, from the young larvae to the foragers, taken from different hives and collected at 2 different time points in the year. And then we analyzed these transcriptomes and we looked into the pattern of expression for all these detoxification genes. Today, I'm only showing what we did or what we obtained in terms of P450 expression patterns. This is a hierarchical clustering of gene expression level, the so-called principal component analysis. And as you can see here, our data nicely separate the different larval stages we collected over time. But you see the cluster of the adults. Although we collected these adults from different compartments in the hive, they did cluster together, so there's no big difference between the adult stages in terms of gene expression. What you see here is it's all the P450s, the entire P450 gene inventory of the honeybee, and you see the expression pattern throughout the life cycle of the honeybee. And many of those P450 have patterns which show a fairly high expression level in the adult stage. So these are obviously those P450s which are involved in the detoxification of xenobiotics, such as, for example, insecticides. So this data is available and will be published soon. Some words on the Arthropod P450 phylogeny, taken this excellent graphics from a paper recently published by Dermo and colleagues in insect biochemistry and molecular biology. And so it's a fantastic paper. And what they did, they analyzed almost 3,000 P450 sequences from arthropods from 31 different species. And they constructed these maximum likelihood trees to show the phylogeny of these different P450 clans and families. I'm showing this because the most important clan in terms of xenobiotic detoxification seems to be CYP3 clan. And you see here these, and this clan also normally includes quite large lineage-specific expansions of P450. And the so-called CYP6 and CYP9 family you'll find in this clan. And also looking at the in the true orthologs, you find, especially in the mitochondrial clan, where you find P450 enzymes, which are involved in the processing of endogenous substrates, such as, for example, juvenile hormone or ecdysone, these hormones involved in the development of these insects. There's not much orthologs you find in this CYP3 clan. What you find in this clan is especially P450s, which are involved in the processing of xenobiotic compounds. That's what you see here, the red dots. Whereas those processing the endogenous compounds, you find in the mitochondrial and CYP2 clan. So what we did, and that is cutting a long story fairly short now in a single slide, work we conducted over almost 3 years. So we were interested in this 2,000-fold difference between imidacloprid and thiacloprid. So we took all this CYP6 and CYP9 subfamily P450s out of the honeybees. And so we cloned these genes and functionally expressed them in a recombinant way in insect cells, in high-5 cells. And then we isolated these individual honeybee P450s, exposed them to thiacloprid, just to find out which one of those hydroxylates thiacloprid. And at the end of the day, it was CYP9Q3 we found as a molecular determinant of neonicotinoid selectivity in honeybees. So you can see here, this is the amount of thiacloprid hydroxy metabolite produced by CYP9Q3. There's a little produced also by CYP9Q2, so CYP9Q2 might also be involved to some extent in the detoxification of thiacloprid. Imidacloprid, on the other hand, is hardly metabolized by this particular P450, despite the close similarity of these chemical structures. We further characterized the CYP9Q3 and confirmed throughout this characterization its role in selectivity. It's, for example, expressed across all life stages. You can see here fairly high expression levels in late larval instars, providing detoxification value and capacity, especially in the adults of honeybees. The major expression sites also make complete sense because it's expressed mainly in the brain, where the nicotinic acetylcholine receptors are sitting, targeted by this neonicotinoid insecticide. And they're also sitting in the Malpighian tubules at fairly high levels. Then we constructed a tool where we ectopically expressed the honeybee CYP9Q3 in Drosophila flies. So that is our transgenic flies expressing a honeybee P450. We have them for CYP9Q3, CYP9Q1, and CYP9Q2. And these transgenic bee-lysed fly lines expressing this honeybee genes turned out to be resistant to thiacloprid, whereas the wild-type flies were highly susceptible to thiacloprid. So flies carrying this honeybee gene survive thiacloprid treatments. So in summary, we identified an exceptional natural mechanism of selectivity in honeybees, which confers resistance to thiacloprid, a neonicotinoid insecticide. The paper has been published in 2018, and so it raised quite some media interest. Then we continued our work because we wanted to expand this knowledge also to other chemical classes of insecticides. In 2018, the EU banned thiamethoxam, imidacloprid, and thiacloprid. And thiacloprid and acetamiprid were exempt from the ban, not because we have shown that they are detoxified by honeybees, but for other reasons. But I'd like to tell you that not all modulators of the insect nicotinic acetylcholine receptor belonging to IRAC mode of action class 4 are neonicotinoids. But many of them, or all of them, are under general suspicion, as they share the same mode of action, under general suspicion to be toxic to honeybees. Although tools we use, such as, for example, cheminformatic analysis, looking into chemical similarity relationships between chemical classes of insecticides, such as the Tanimoto indices you see here, these are all different classes of chemistry acting at the orthosteric site of the nicotinic acetylcholine receptor. Here you see the large neonicotinoid cluster, but you see also other clusters grouped in IRAC mode of action group 4, such as, for example, the butenolide cluster. The butenolide cluster, here you find an insecticide which has been recently launched, fluopyradiferone. It's called Sivanto. And it's 1,000-fold less acutely toxic to honeybees after topical application compared to imidacloprid. It's 300-fold less toxic to honeybees after oral application. And it only 6-fold less lower affinity to nicotinic acetylcholine receptors. Another compound out of this sulfoxamine cluster, which is also separate from the neonicotinoid insecticides, is closer, sulfoxaflor, which is a compound also acting on the nicotinic acetylcholine receptor. So these compounds are chemically different from neonicotinoid insecticides. Therefore, they have been separated from this chemical class. I'd like to give you one example because I'm always asked, so that's very difficult to see the difference between these chemical classes. And Einstein already, there's a quote from Einstein saying that everything should be made as simple as possible, but not simpler. Certain groups, I would say, take this, and in 2016, there was a paper published from a European network asking the question if sulfoxaflor and fluopyradiferone, are these neonicotinoids or not? And from a chemical perspective, of course, it's always difficult for a layman to differentiate chemical structures. And they took the example, several characteristics, and compared sulfoxaflor and fluopyradiferone with other neonicotinoid, they call them other neonicotinoid insecticides. Do they bind to insect nicotinic acetylcholine receptors? It's all yes. Do they function in a systemic way? It's all yes. Are they toxic to bees? It's all yes. So, and the conclusion out of this is that their properties clearly show that they should be classified as neonicotinoid insecticides. If I can also ask a different question to you. Would you say, for example, cats and dogs, bears or not? If I would follow the logic of this, what's listed here, I would say they are all mammals, they are all carnivores, and cats and dogs also have 4 legs, similar to bears. So the conclusion is that their properties clearly show that they should be classified as bears. I would say that's nonsense, and but it's much more easier to see the difference between these 3 guys than the 3 chemical structures. And we use this approach, the toxicogenomics approach, to explain the safety profile of the butenolide insecticide, fluopyradiferone. So first of all, we looked into the metabolism of fluopyradiferone in honeybees, which have been exposed to radiolabeled fluopyradiferone. And it turned out that we found a number of metabolites which are based on the oxidative degradation of certain substructures in this molecule. And then we used our cytochrome P450 expression library from the honeybee and tested the 27 different P450 genes for their ability to detoxify fluopyradiferone. And it turned out that 3 P450s, surprise, surprise, CYP9Q3 and CYP9Q2, again, are involved in the detoxification of fluopyradiferone, but also another one, which has not been shown to be involved in any detoxification yet, is CYP6AQ1. And then we analyzed the different metabolites which were obtained by the different P450s. And what we show here is that this compound is metabolized by 3 different P450s, and that is the metabolic fate of fluopyradiferone in the honeybees. So our molecular approach helped us to understand the metabolism of fluopyradiferone in honeybees. And the major metabolite is the difluoroethanamine here. Then we used the transgenic flies. This time we used a different version from those we used for the thiacloprid. And so in this particular case, the P450 genes from the honeybee are expressed under heat shock. So we use 2 fly lines, a driver line with a GAL4 a transcription factor, which is under HSP or heat shock protein 70 promoter. And we used another line which has a so-called upstream activating sequence and a P450 of interest, in this case, CYP9Q2 and CYP9Q3 of the honeybee. These flies were crossed, and if these flies are heated, GAL4 is expressed, binds to this activating sequence, and then the P450 is expressed. And we were able to show that flies expressing CYP9Q2 and CYP9Q3 become more resistant to fluopyradiferone. That is the dose response curve here, the squares for the CYP9Q3 transgenic flies compared to the wild type flies, which are the empty or flies which are not expressing any honeybee P450. So these transgenic flies are very nice tools to check, for example, if certain enzymes are involved, a year-round tool, if you want, in those climates where you do not have access to honeybees year-round, where you can check if these enzymes are involved in the detoxification or not. Another important thing, of course, we call this mechanistic risk assessment or genomic risk assessment, but you always need to do bioassays and check. In this case, the authorities, of course, asked for what is the toxicity of the metabolites we obtained for fluopyradiferone. And what we can say that all these metabolites produced in honeybees are practically non-toxic to honeybees. If you compare this, for example, to the major metabolite of imidacloprid, which is imidacloprid hydroxy, it still remains, even though it's a metabolite, it remains highly toxic to honeybees. So the main metabolites resulting from P450-mediated detoxification in honeybees do not bind to the nicotinic acetylcholine receptor, and very important after contact and oral exposure. Another topic we checked was what about synergistic effects? Can we determine synergistic effects by using molecular tools? Synergistic effects between pesticides are an issue of major concern from an ecotoxicological perspective. And you see here data I've taken from this paper where the combination of thiacloprid and prochloras led to high levels of synergism and toxicity. The same has been published 2 years ago for fluopyradiferone. In this paper by Tosi and Ni, showing that propiconazole synergized the toxicity of fluopyradiferone. We also checked this under laboratory conditions, and what you see here, the black line is the dose response curve for thiacloprid. And if you combine thiacloprid with propiconazole or prochloras, you're shifting this dose response line to the left, and it becomes much more toxic. So it's highly synergistic. Whereas other azole fungicides, such as, for example, thiaconazole, is not synergizing the efficacy of thiacloprid. We observed the same for acetamiprid, and we also tested azoxystrobin, and azoxystrobin is also not synergistic in combination with this neonicotinoid insecticide, whereas the two others, prochloras and propiconazole, are highly synergistic. The same is true for fluopyradiferone. Highly synergistic, these two compounds, propiconazole and prochloras. Then we developed the so-called fluorescent probe assay, employing recombinantly expressed CYP9Q3 to measure pesticide P450 interaction. Almost similar to what's done in the pharmaceutical industry to check drug-drug interaction with human P450s. So we checked for an appropriate substrate and identified benzoxyfluorocoumarine as an appropriate substrate for CYP9Q3. And this substrate is metabolized to this product here, which is hydroxycoumarine. And this coumarine emits at a wavelength of 410 nanometers. So it can be used in fluorescent assays. You can measure the fluorescence. And this is also a test system you can conduct in 384 well plate formats, so a very high throughput. And this is a typical control result here, where you have increasing fluorescence with increasing concentrations of the product used. If you are now adding a pesticide to this reaction, and the pesticide interacts with the same binding site, with the same catalytic site as a BFC substrate, you would see a depletion or decrease in fluorescence. And that's exactly what we observed when we are adding increasing concentrations of thiacloprid to this reaction. You see that the fluorescence signal is going down. In other words, thiacloprid and BFC are competing for the same catalytic site in this enzyme. So this could give you an indication that thiacloprid might be detoxified by this P450. The same is observed for acetamiprid, whereas we did not observe anything for imidacloprid. So imidacloprid does not compete with BFC for the active site. And interesting enough, we showed that nanomolar concentrations of prochloras and propiconazole inhibit this enzyme by using this particular assay. And the strong inhibition of CYP9Q3 by these two azole fungicides at very low concentrations explains at the molecular level the observed in vivo synergism in combination with thiacloprid and fluorodeferol. We did not find an inhibition of by prothioconazole, indicating also and confirming what has been observed in vivo. As you can see here, also fluorodeferol at increasing concentration is decreasing the fluorescence signal. In other words, also fluorodeferol directly interacts with CYP9Q3. And if these two P450s are inhibited, this may cause synergism. If this also causes synergism under field conditions, need to be tested. I can only check this under laboratory conditions. So there's a further read if you are interested in this. I think that's, and it has been taken up thanks to Scott McGart. He wrote a very nice article about this paper we published in Environmental International on this mechanistic risk assessment at the molecular level. And so just for you, if you're interested. So bee pollinator toxicogenomics, we also expanded our approach to species beyond Western honeybees. And we identified orthologs such as CYP9BU1 and BU2 in the red mason bee, which are also detoxifying both thiacloprid and fluorodeferol. We also identified, and this is work done in collaboration with Exeter University and Rosmestad Research, CYP9Q4 to 6 from the bumblebee, also in a position to degrade thiacloprid and other insecticides. So the question is it an evolutionary conserved mechanism we discovered here in the CYP9Q-like enzyme family among bee pollinators? We looked into the Megachile rotundata, which is the alfalfa leafcutter bee. It's also a managed bee pollinator, which is used in alfalfa seed production in the US, for example. And what we observed is that they are lacking a CYP9Q-like enzyme. And that is a branch here in the phylogenetic analysis where you don't see, I would say, an ortholog. In all other different subfamily and branches, you see an ortholog of Megachile to the honeybee. So we know that insecticides such as thiacloprid, taufluvalinate, and fluorodeferol are considered practically non-toxic against honeybees. But these compounds turned out to be highly toxic. Even taufluvalinate, which is known as an in-hive medication against varroa mites in honeybee hives, is becoming highly toxic. And this could be due to the lack of the CYP9Q-like branch. And that has been published recently in Nature Ecology and Evolution. We picked this up because there is a growing interest in other bee species for pesticide risk assessment. We thought, okay, there's 20,000 different bee species, so we need a more focused approach. So what can we do at the molecular and genomic level to address this question about selectivity for many more species than just bumblebees, red mason bees, Megachile, and honeybees? And so there was a project which is has been established here on the phylogenomic tracking of CYP9Q orthologs, and we checked them as selectivity determinants, conserved maybe across bee families. And that's work done by a PhD student working in my group, Julian Haas. He also published some of the work I presented earlier. So what we did, we looked into transcriptomic and genomic sequences available publicly. And we did 79 assemblies from 64 unique species, and coming from the 6 major families. I have to apologize for the taxonomists amongst us, because we have not tested this particular group here or family here, because that is a very small family of bees, only 21 species restricted to Australia. But for the other main families, we extracted P450, and we did the phylogeny and found out that almost all of those species have a CYP9Q ortholog present in their genome or assembled transcriptome. And that's what you see. That is the setup here. We have the different CYP9Q enzymes, 9Q-like enzymes extracted. And then we functionally expressed all these CYP genes along with cytochrome P450 reductase in high 5 cells using a baculovirus expression system, and characterized their catalytic capacity. And here you see the coumarin fluorescent probe substrate profile of the recombinantly expressed CYP9Q-like P450s from 20 different bee species. And you see most of the P450s we functionally expressed show us good preference for coumarin substrates. The more red it is, the better is the catalytic capacity to transform these precursors into hydroxycoumarin. So almost all of these P450s show good activity. So that means we were successful in expressing them, except this P450 from Bombus impatiens. But there was another one which was then successfully expressed, belonging to the same family. And that is work which is in preparation for publication. We also looked into the pattern of fluorodiferone metabolism for all these different P450s. And we were interested, is this a conserved detoxification principle across species from different families? And what we observed, indeed, all these P450s we functionally expressed show a similar pattern of metabolization as the honeybee. So these are obviously orthologs capable of doing the same job as CYP9Q3 in the honeybee. That's what you see here. The extent is different, but all these enzymes, I would say, metabolize fluorodiferone. And the major metabolite also here is this difluoroethanamine metabolite in red. So one example just here, so we also looked into the alkali bee, which is Nomia melanderi, and we also found a CYP9Q-like ortholog, which degrades both thiacloprid and fluorodiferone in this bee, which is used as a managed pollinator in the Western US in alfalfa pollination. And so this candidate CYP9Q3 ortholog was identified. It shows a coumarin spectrum similar to CYP9Q3, and it was also shown to metabolize thiacloprid to hydroxythiacloprid. And we have observed the same pattern of metabolization for fluorodiferone with this Nomia CYP9Q-like enzyme. So this could be an indicator that if you are testing these compounds in a bioassay, an acute toxicity bioassay, there's no bioassay available for this bee species. But we can predict from this particular result, at least from the molecular level, that these bees are in a position to detoxify those compounds. So it could act as a predictor. So and that is at the end the goal of our studies, to phylogenomically track this detoxification. How conserved is it among different tribes? And so we did this for Apides, by far the most important family, with more than 5,700 species. And we found them also in different families, the capability using our fluorescence depletion assay, if you want, that thiacloprid is decreasing the activity of this enzyme with the substrate, in this case, BOMFC. We also found this for the Coletidae family for species we selected, and also for the Halictidae. You see here that's very impressive, the suppression of the fluorescence signal in this particular species. And maybe we can really define nodes in the phylogenetic tree which help us to say, okay, everything that's below this particular node would, I would say, help us to say these bee species belonging to these subfamilies are very likely in a position to detoxify these compounds because they have a P450, which is orthologous to CYP9Q3 of the honeybee. We also did some in silico screening for signature amino acids and looked into computational modeling. And that is work which is, I have to say, where we failed so far to find really principles which could give us a tool in our hands where we are able to predict this via an in silico approach, because we have checked. There are not too many P450s, and there are much more structures which need to be checked. And it's also hampered by the fact that we do not have a crystal structure of any insect P450 yet, where we can do homology modeling, which is not based on human CYP3A4. And this is work we are doing in collaboration with Carly Loon at the EPA in the US. So there has been a paper, now I'm coming to the end of my presentation, there has been a paper published in Transgenic Ecology and Evolution, and here the authors asked, that was by Lopez Osorio and Wurm, and they asked that what's necessary are high-resolution approaches from molecular medicine, which can provide unparalleled insight into organismal physiology and health, and applying these approaches, and that's what we did with our approach. We applied these approaches, which aims to significantly improve the efficiency and sensitivity of pesticide research and evaluation, because all these tools can be used also to inform pesticide risk assessment in bee pollinators. Just a brief summary of this approach. So toxicogenomic and mechanistic studies help to identify and understand evolutionary conserved molecular principles of selectivity among bee pollinators. And I think that's what we have shown, where we looked into the orthologs from many, many different species. Bee toxicogenomics and phylogenomics helps to inform and explain issues of regulatory concern at the molecular level. For example, synergist, how are pesticide interactions, and how do they affect P450s and detoxification. The phylogenomic tracking of selectivity and identification of conserved detoxification gene families may help to cluster tribe-linked sensitivity issues. So that's what I've shown in one of the earlier slides. So honeybee CYP9Q like orthologs were found in all 6 bee families, which is good news, in fact. So at least for those compounds which are actively detoxified by these P450s. Biochemical high-throughput screening methods based on bee P450s help to predict speed and extent of metabolic clearance of candidate insecticides in early discovery. So it's a useful method. And we are, of course, striving for an improved science-based communication with regulatory authorities worldwide by using such an approach. Last but not least, I have to acknowledge, of course, my fantastic team here at Bayer in Monheim. So for me today, it's more a dinner and learn webinar. So it's always late in the evening. And so there are many people I have to thank, especially Julian, who did a lot of work on the bee toxicogenomics, but also other PhD students in my team earlier, such as, for example, Marian and several postdocs, which provided insights into this topic. And my long-term collaborator, Chris Buss, and his team at Exeter University, as well as Lynn Field and her team at Rosenstedt Research, John Bontas and his team at IMBB, Crete. And yeah, I'd like to thank you for your attention. That's all I have for you today. Thank you.
Wow, great. Thank you so much, Ralf. This is just an amazing talk. I was blown away by all of the
Thank you.
the beautiful combination of the chemistry, the biology, the genomics, all the different tools that you use to really try to find out what's going on inside the bees, and then being able to extend that knowledge and make some conclusions about other species around the world. And so congratulations on this wonderful work. And so I have a few questions in the Q&A tool. Will Ridley asks, I'm interested in the conduct of the LD50 test for honeybees using insecticides, route of exposure, bees per test, number of tests. Also, are bumblebees included in the insecticide evaluation? I think you kind of answered that during your talk, but perhaps you could.
Yes, I think that
retest
there are guidelines and there are EPO standards. We have to OECD standards. We have to follow, and it's clearly explained how many bees, how many replicates, how you should dissolve your insecticides, to use a formulated material, technical material, temperature, what is the at what time points are you scoring for motility. That's all outlined in these OECD standard procedures, and that's what we are normally following. No such standard procedures are yet available for example, solitary wild bee species, which may come also onto the plate as a mandatory species for regulatory purposes over the next years.
One interesting question from Laura McHenry. Thank you for the interesting talk. Since the vast majority of bee species you looked at have the CY9Q orthologs, how might we explain the lack of an ortholog in the Megachilrotundata?
Yeah, the alpha. Yeah. So that is a very interesting and a very good question. So that's because that is what we are now looking into, because we are seeking for bee species. It's not that Megachile family is lacking CYP9Q orthologs per se, because Osmia is also Megachilidae. It's the same family, but they have an ortholog. So now we are looking into other genomes, honeybee or other bee genomes from different families. If there are also other cases where this particular orthologs are lacking, it's quite a lot of work. But I think the positive news is that many bee families, or all the species we tested out of 6 different major bee families, have orthologs. And to put this into a risk context, I think it's good news that there's principally the ability. What we need to do normally, I would say, you have to correlate this predictive thing and the molecular work by doing bioassays, acute toxicity bioassays. But many of the species are not accessible or available for toxicity tests, and that is, I think, the very difficult part of the work.
Yes, because I think the point is that regulators have concerns that species that are not actively tested, that they had a concern that maybe they weren't being protective of those species, correct?
It's correct, but maybe our approach can help to lower these concerns a little bit, because if we say, okay, they have a detoxification enzyme, which is in principle in a position to detoxify those insecticides we tested, and they may not detoxify all insecticides, but at least you can use these molecular tools to check which chemotypes are detoxified by these enzymes.
Fantastic. Another question is, does the route of exposure, oral versus contact, impact toxicity?
Yes, for sure, yes. That is, some of the compounds do not penetrate very well the insect cuticle, which is good for the insect or good for the bee, but they are taken up orally, and so there are differences. There are examples where you have insecticides which are practically non-toxic via the contact route, but they are moderately or even highly toxic via the oral route. So there are differences, yes.
Okay. Any thoughts as applying this approach to try and increase pest spectrum, or at least enhance understanding of basis for species selectivity among pests?
Yeah, give me another 30 years and I would really, yeah, of course, this approach, the dimension is incredible. Yeah? You can think about also looking into pest selectivity, but with pest selectivity is often due to differences in the target sites. Now, that is, if you compare, for example, aphids and moth species, and the reason why certain insecticides are inactive in hemipterans or aphid species is because the target site is slightly different, or the binding site for the insecticide. Yeah? It's, but it can also be detoxification mechanisms. It's known. Yeah?
Okay. How many CYP 450s are closely regulated? In many CYP 450s, they're closely regulated by the circadian rhythm. Have you seen any circadian toxic pattern for CYP P9 in mellifera?
No, I haven't. And I haven't. If I say no, I haven't, I would say we did not check this. Yeah.
Okay. Okay. Let's see. Would it be possible to explain bee safety for flupyrimin on the same grounds? Why is it still difficult to design insecticides, bee-safe insecticides, despite so much molecular understanding?
Yeah, I think so. What we did with flupyradiferone, for example, that we used the same approach to find out which P450s are involved, and what we observed that it seems to be that CYP9Q2, 3 in the honeybee are quite promiscuous in terms of the substrates they accept, and their catalytic capacity is quite high because I cited a paper where it has been shown that cumafos, an organophosphate, taufluvalinate, and pyrethroids. We have a butenolide insecticide. We have a neonicotinoid insecticide. And we have also a work ongoing where we are showing that other chemical classes of insecticide are also detoxified by the CYP9Q family. So this is really interesting, but it remains difficult to identify why do they have this catalytic capacity, because we are lacking a crystal structure. You can do docking in silico with the compounds and can say, okay, that might be a residue on the chemical structure which affects binding to the P450 or not. Unless you have no real co-crystallization with an insecticide and an insect P450, it's best CYP9Q3, of course, then it remains difficult. Yeah? And so that is the reason. It can be used, it can be introduced as an early screening strategy, of course, but you never know. If it's not metabolized by CYP9Q3, it does not mean anything. There might be other enzymes.
Well, I think because we've already gone over time, we're going to wrap things up. And I'll look, many people are saying great talk and congratulating you on your presentation. So I think definitely I learned a lot about this topic, and hopefully the attendees did as well. And hopefully in another few years you can come back and tell us all about the new discoveries that you made in this area. It seems like you've uncovered sort of a new line of research that could be huge over time.
We keep the ball rolling, and so that's I think that's a good news. So we are still working on this topic. Yeah.
Oh yeah, definitely. Well, thank you so much, Ralf, for being here today, and we really appreciate you putting together such a wonderful talk and presenting today as our last webinar of 2021. And so very exciting, and thanks everyone for joining, and hopefully we'll see you. Our next webinar is scheduled for February of next year, and we'll be talking about cannabis. So it should be very interesting. So, okay.
Thank you.
Bye-bye, everybody. Have a good day.